Hi, everyone. Welcome and thank you for joining the fourth of the six-part Particle Classroom series, System Verification. My name is Julie Chen Nguyen, and I'm going to facilitate today. All right, so it's my pleasure to officially introduce you, our speaker, Dr. Jeff Bodycomb. Dr. Bodycomb is our product line manager for particle characterization. He received his PhD in material science and engineering from Rutgers University and has years of experience focusing on dynamic light scattering, like laser diffraction, imaging analysis. He is especially interested in sources of error and measurement, scattering techniques, and microscopy. So I can only imagine that this webinar on system verification is somewhat a passion of yours. All right, Jeff, I don't know. I'm going to go ahead and pass the ball to you. Please go ahead and share your screen. I'll mute myself.
Okay. Well, thank you for the introduction, Julie. Like she said, this is Jeff Bodycomb, and I'm here to talk about system verification. So let's get to it. This is going to be a fairly short webinar. So let me talk about what I'm going to do. And most of the time, system verification is pretty boring, and and I say that in a good way. You're really trying to establish, is my analyzer working, which comes down to, is my analyzer clean, and to some extent, is your procedure okay? I'll comment on each of those things, procedure a little bit later on. Really, this is not asking, is my unknown sample okay, or am I measuring an unknown properly? That's really the province of method development. But before you really start working on unknown samples, you want to make sure that your analyzer works. And I'll give you an example from one of our customers in the pharma industry. They would verify their analyzer before every set of measurements for drug release. And it turns out they were doing this because they were sharing the analyzer with the R&D group. And sometimes the R&D group would leave extra particles in the analyzer. And this was one way to make sure that their measurements on the drug products were correct and no one messed things up in between production campaigns. So, like I said, most of the time this is pretty boring. It's a great way to have confidence in your data or where you're getting started. A couple words or definitions. So there's the question of calibration versus verification. And the idea generally when people say calibration is you put a standard sample into your instrument, you adjust the instrument response to show the expected result. Examples would be particle counters. Another example would be if you're checking, confirming the magnification of a microscope, you put a reticle in and you'll note how many microns per pixel that you're measuring. Verification is the term I like to use when I put in a standard sample and I check to see if the response is within the accepted range. And that's really what I'm talking about today. Examples of this are dynamic light scattering, laser diffraction. If you look at ISO 13320, on calibration, laser diffraction systems are generally based on first principles. So calibration in the strict sense I mentioned above is not required, but you do still need to confirm correct operation by a validation procedure. This comes down to the geometry in the instrument and the wavelength of light you use is pretty well fixed. It's not going to go anywhere. And if you put in a material with known particle size and you get the wrong answer, that means that either you have a procedure problem or your analyzer is broken. So this is what I mean by it being boring. If you get the right answer, you just move on your way and run your unknowns. And if you don't, it can get pretty exciting. So again, verification, 13320 should we prefer that validation be made with a certified or standard reference material acceptable to the practice of the end users industries. This is kind of a way of saying that you'll want to use standards that your customers will expect and trust, or whoever is receiving the data. 13320 recommends, and so do I, that you have reference materials with a known distribution having a range of sizes over one decade, and generally spherical particles. The reason I like spherical particles is because with a hard sphere, it's the only sort of particle where you get the same answer from every analytical technique. Note, one, this implies using a polydisperse standard. Two, most people still use monodisperse standards, and most polydisperse standards are not don't have that broad a size range within the standard. Let's talk about monodisperse standards. They're very popular. It's a very narrow size distribution. All of your spheres are the same size. It tests the central point, no data on D10 or D90. You can buy them in sizes from 20 nanometers up to 1 millimeter. Generally, they're suspensions, although larger ones will appear as dry powders. They're easy to use, and they're relatively inexpensive. So that's a good argument for using them pretty regularly. It's not going to cost you that much. It verifies generally your instrument optics and your instrument cleanliness. So if you have a 1 micron standard and you put it in your analyzer and you measure a broad distribution with a peak at 2 microns, I'm probably going to suggest that you clean your analyzer and clean your O-rings, because that's probably what your problem is. But even after cleaning, if you still have problems, it suggests there's an issue with the instrument optics. So why do I mention optics only? Well, if I take 1 micron particles and put it into suspension, and my pump is not pumping very well, I'm still going to have 1 micron particles sitting in suspension, and I'll have a very nice measurement result of 1 micron. And I'll smile and say, hey, my analyzer is working. Another case is if you have particles that are not being well suspended, they're all floating on top. If you even get just a few into your measurement area, you'll get a good result. You can say, hey, my analyzer is working. And so with these monodisperse standards, I'll know that my optics work, but I could very well have a pumping or mixing problem that does not come up, because all I need to do to make the measurement give the right answer is have some mixing and some pumping. It doesn't need to be particularly vigorous. And then when I go to measure an unknown sample with a range of sizes, where I need to make sure that all the material is circulated, I'll start to have problems. There are no real official pass-fail criteria, so generally you'll make your own around what you think your analyzer can do and what you need. Quick comment, there isn't a lot of profit in making a pass-fail criteria that is tighter than you need it, because as your criteria get tighter, you have to be much more careful with your measurements, and that means that as you need more and more care in measurements, the measurements become more expensive. Generally, we like to measure 3 different particle sizes as monodisperse standards: 0.1, 1, and 100 micron polystyrene latex. And those are generally kind of quick checks of instrument operation. And then we'll go back and measure a polydisperse glass bead standard. There are a huge number of sizes available. There's a list on the right of sizes available from Thermo, which is really the old Duke Scientific. You only really need a couple particle sizes to cover all of your detectors. And just as a comment, larger particle sizes tend to be more expensive. There's no real rule that says you need to measure near your actual sample size. You really need to measure a sample that excites most of your instrument detectors to check out your optics. By the way, our processes say plus or minus 5% of the certified value on a measurement of a monodisperse standard. So polydisperse standards are much more interesting. They have a defined or certified distribution, and ISO and USP both have defined procedures for pass-fail criteria. And you can purchase them from White House Scientific or directly from the US government, NIST. This really tests the entire instrument, and you can pick up really what's happening in your system performance. And that's all for the good. They can be more troublesome to work with, and they are certainly a whole lot more expensive. So you're talking with the monodisperse standards, you're talking about, I don't know, on the range of $10 a test, and then 10 times that for the polydisperse standards, as a real rough approximation. Yeah. So White House Scientific has a good range of sizes. If you get the smallest of their size ranges, the 0.1 to 1 micron, then you're going to be testing your ability to disperse the particles as well, which may be what you want or may not. But be aware that with these standards, you're going to get into questions of dispersion and dispersibility, and we do get into trouble with that sometimes I found lot to lot, some beads tend to spread out better than others. I can't figure out why. So that's kind of bad news. The good news is you can buy in a proper quantity for a given sampler. And so we don't have any sub-sampling concerns. So if you go to White House, you can buy these tiny little tubes of glass beads, and you just dump the entire tube into the system and make your measurement, which means you don't have to worry about super careful riffling to ensure that every bottle has exactly the same distribution of sizes. White House has basically done that for you. And so anyway, I'm kind of a fan of that. So if you look at some of these older prices, a 10-bottle set can be $400 to $194. So that's $40 a bottle. So we don't tend to use that. We use 202, which is probably $20 a bottle. And this is an old price list. This is really your only option for dry powder feeders because it's already split for you. And there's really no point in measuring more than one size range. So we'll pick just one of these for instrument verification, unless we think something special is going on. So let's look at NIST 1003C. The size distribution is shown on the left here. And kind of what I point out is that there's electrozone sensing and laser diffraction, and you have two different distribution curves. And then actually NIST and White House both use multiple techniques to characterize the particles. And so you'll get somewhat different results for different techniques, and that will start feeding into your uncertainty. You'll also see a lot of different uncertainty expressions in these certified diameters, which, as Julie knows, I'll talk about over lunch, but probably save you guys from that. So how do we go and test it? Well, I test samples 3 times for repeatability, and really I call this reproducibility. So there are 2 ways to do this. One is you put a sample into the system, you hit measure, you hit measure, hit measure. If you do that 3 times in a row, you're really testing your system repeatability. This is a important verification for your system when you can do it, because really you're looking at how good is the system. If you do independent measurements where you prepare, measure, drain, and repeat, then you're really checking reproducibility, which is your bottle-to-bottle variation, as well as your system variations. I don't like to do that as much if I'm trying to confirm analyzer operation. I have to do it with dry powders, but with liquid suspensions, I do not. And if you ever listen to me talk about method development, I differentiate between repeatability, put a sample in, measure 3 times, and reproducibility, clean the system, measure again. Very analogous here. And then you're going to look at the mean and coefficient of variation for these repeats for D10, D50, and D90. And if you look at ISO 13320, we want D50, or the median, coefficient variation less than 3%, and D10, D90, coefficient variation less than 5%. So I should be getting the same answer in these 3 repeats. We'll double limits below 10 microns because those measurements tend to be more tricky. You'll note the text reads X10, X50, X90. The legend is that the Germans prefer the X and they supervise most of the writing, and most people in the world tend to use the D for diameter. There's a statement on validation, and which is kind of a restatement of what I just said, that the mean value deviates less than 3% from the range of values and less than 5% for D10 and D90. Again, some precautions. Validate with respect to precision, accuracy, at regular time intervals. So as you think about your analyses, you want to decide how often to check your analyzer performance. If it's very, very critical, or if you know that some nonsense might be going on, then you might check more often. If it's less critical, then you'll check less often, or once you establish the instrument operation is stable, you'll start checking less often. So for the verification test, you measure standard 3 times, calculate the mean value of your D10, D50, D90 values for your accuracy, and then calculate your coefficient of variation for the precision. So D50 has to be within 3% of the range, and D10 and D90 within 5% of the range of values. So what is this certified range of values, and how do I think about using this 3% on D50? Well, on your right, I show this certified diameter values off of the NIST table. You'll see similar tables from White House. And I'm really interested in 3 columns: the mass fraction, diameter, and expanded uncertainty. And if you look at the guides on uncertainties, expanded uncertainty tends to be at 95% confidence. So you'll take a standard uncertainty and multiply by 2. If you look at the center columns, that's how you build up this expanded uncertainty. Anyway, you can see that D50 of this is 32.1 microns. With expanded uncertainty 1 micron. That says that really D50 on this bottle could be 31.1 micron or 33.1 micron. And about 95% of the time, it's going to fall within that range, or 5% it won't. So 1 time out of 20, you're not going to get the right answer, just because of the uncertainty going from bottle to bottle. So when I calculate my pass-fail, the first thing I don't want to do, so this is D10 at 21.8 microns on the table at the bottom. Well, if I just say that's plus or minus 5%, I go from 21.8 times 0.95 is 20.7 microns, up to 21.8 times 1.05, it's 22.9 microns. So I have a range of 20.7 to 22.9 microns. The problem with that is that you have a bottle uncertainty and you haven't accounted for that. So the way to think about this is say, look, my lower limits can be set by the lower limit of my bottle, 21.8 micron minus 1 micron from the expanded uncertainty, times 0.95 is 19.8 microns. And then 21.8 plus 1 micron times 1.05 is almost 24 microns. So your allowable range is actually 19.8 to 24 microns with some rounding. The reason we want to account for the bottle is the instrument can't control what's happened with the bottle and can't control what's happened with the bottle. So you can imagine a case where your bottle to bottle variation is quite large compared to the instrument variations, and you'll just keep getting trapped by bottle to bottle variations. So here's a worked examples with the D50 and D90, giving ranges of, for D50 of 30.2 to 34 microns, and D90 range to 38.4 to 44.5 microns. This isn't really the whole decade that ISO 13320 wants to get to. I think that goal is more aspirational than something you can execute today. But hopefully with better developed standards, we'll get there. The challenge with these broad size distributions, and the challenge that White House is taking care of for me really, is that the broader your size distribution, the more carefully you have to split your sample to ensure that every bottle has exactly the same size distribution. If all your particles are the same size, you can take off the bottom, you can take off the top, it doesn't matter, they're all the same size. If there's a range of sizes and there's any segregation, you have to be very careful about how you separate materials. So, a couple of comments. Pay attention to your procedures. I had a customer who was measuring 100 micron polystyrene beads, and they're like, oh, I'm using so much material, running out, and they need a lot of material for a test, and the scattering signal is very, very weak. Well, 100 microns, you should be scattering quite nicely, and you shouldn't be using that much material. And so I looked at what they were doing, and you can look in the sample bath and say that the beads were floating to the top of the cup because their agitation was set too low. And so once we set the agitation higher, we can ensure the beads were well entrained. I wanted to consider some surfactant, but because of the way their internal procedures worked, that would be quite a lot of grief for them paperwork-wise, and so they didn't do that. But that would have been the next step, is to really add something to ensure the particles were completely wetted. So here's some example data with PS202 on a 960, our laser diffraction analyzer. I have D10, D50, D90 with a standard value of 9, 13, and 20 microns, and the uncertainties about 1 micron, and notes on the allowable error range. And then you'll see our calculated lower and upper limits and the measure results, which sit right in between. So that makes me feel good. Everything's working. PS225 from 5350 microns. Again, a very similar story. So this is 94 micron up to 240 micron. we can calculate the lower and upper limits and see that the measured results are right in between. So really, if I get a number that's between this lower and upper limit, and I've done the calculation right, I declare victory and move on to real samples. Often your uncertainties in regular samples are coming from sampling or dispersion, and these kind of differences that we're seeing with the standards don't matter too much. PS 181, 0.1 to 1 micron. This is where you're going to have to really watch your dispersion. This is where you're going to have to make sure you might consider adding some surfactant as you disperse these beads. This is 0.4 microns at D10 up to 1.1 micron at D90. And the measure results 0.28 to 0.44. So we're a little on the high side, but we're still well within spec. 0.65, this is where we expect it. And D90, again, pretty close to where we expect it. Yeah, so this table isn't too interesting. I guess really take home on this is to remind you, think about surfactants. You're allowed to use them. You don't want to pick a surfactant that's so large it significantly affects particle size, but I can't think of anything that's going to bother even this 360 nanometer particle. That's a common surfactant. Quick comment, USP 429 is used only in the pharmaceutical industry and is based on ISO 13320. It has broader limits for reproducibility, but the same limits for verification. So if you're in pharma, you might go and look at this standard in addition to 13320. Okay, I just had a question, which, yeah, what if the measure result does not fall within the upper and lower limit? And I'm gonna go ahead and talk about this. If the measurement result doesn't fall within the upper and lower limits, there are kind of three things that could be. The first is you made a calculation error on the upper and lower limit. That's the least common. The most common is that there was a procedural error in your measurement. For example, if we go back to this 0.1 to 1 micron standard, it could be that you did not sonicate long enough, or you did not add surfactant, and so you just weren't able to break up the small aggregates of these 0.1 micron glass beads. And that does happen. I've seen some bottles of these smaller standards that just disperse beautifully right away, and some bottles that are a little bit more resistant. So that's an issue. It could be that you have something in your analyzer that is adding particles to the system. For example, if you use something particularly sticky and didn't clean it out very well, or if your colleagues did use something sticky and didn't clean it out, you could be seeing extra particles in your measurement, and you need to clean the system. And finally, if you really can't get it taken care of, then it's time to figure out what's wrong with your analyzer. But the first place I would look is the calculation, because that's easiest. Sometimes people grab the wrong bottle of standard when making a measurement, and that's pretty easy to fix. Sometimes you make a math blunder, and that's easy to fix. Sometimes it's procedural, rerun it, everything's working right, it just means you missed a step. And sometimes you do have an issue with your analyzer and you need to go figure out what's wrong, whether your laser is failing. That will tend to, if your laser is failing, particularly on older analyzers, your distribution's tails will tend to go out more than the center, and you'll pick that up more with these polydisperse standards. But the first place I always look is in the procedures and talk a customer through their procedures, because you don't want a service visit if it's just kind of how you're approaching the measurement. The LA960 does have tools for automating the calculation and the verification. So report, yes, it's okay, or no, it's out of range. So look at, say, D50, D10, D90. Okay, no one's asking about aqueous versus solvent yet. So everything I talked about so far is been running stuff in water. And then what if you're running solvent? So the common procedure is to switch over to water, test, and then go back to solvent. But let's say you're using an organic solvent, say toluene, that's toluene water immiscible, which means you're going to have some sort of emulsion if you just try and switch directly over. So you'll need to use an intermediate solvent to get rid of your nonpolar solvent to switch to water, and that might be something like THF, might be something like alcohol, and then you'll have to really rinse all that out. So you really have to watch your background and make sure that you don't have extra immiscible solvent floating around, because that's going to be a problem. So then how do you know it's clean? And one approach has been, well, test the system until it passes. This example of glass beads in IPA versus hexane, and kind of scratching your head wondering, I'm not passing in hexane, why would that be? One thing is the refractive index of hexane is very close to that of glass, and so you're not going to get very much scattering. So you just don't have much signal to work with. But when you work in IPA, it works quite well. So organic solvents tend to swell your polystyrene beads. They tend to have refractive indices very close to glass, which means that you don't get very much scattering signal. And that's going to make those measurements more challenging, which may push you to switch over to water, which leads to hole cleaning challenges as you switch back and forth. So I just want to warn you, you do have to think about those sorts of issues. This was done in the LA 920. I should go back and do it on 960 just to see if the improved sensitivity will get me better work with hexane. I kind of promised a short webinar, and it's going to be short. In conclusion or concluding comments, verify your system on a regular basis. It should be risk-based. If you're running a huge plan on particle size, you may do a daily check. And your daily check might be with a monodisperse standard, with a monthly check with the broader, more expensive standards. If you are using this for your research and you only need critical measurements a few times a year, then you might go to a full check every year. Recommend everybody switch to using these polydisperse standards, which also test your samplers. Unfortunately, they also test your bank account. That's what people tell me. And just be aware of the risk that when you use monodisperse standards. I think it's great to have something you could check real quickly every day. I carry 1 micron polystyrene latex when I'm traveling, just as a quick test on the system. But you want to do the broader standards on some sort of basis so you have a stronger paper trail and you're more aware of what else could be going on with your system. So I'd like to thank you for your time and open the floor, the electronic floor, for questions.
Well, thank you, Jeff, for the excellent webinar. I do have one that came in during registration, so I'll probably rephrase. This is, what would be expected of the operator when it comes to system verification on particle size analysis?
So, so the person in front of the instrument, there's two things the operator needs to do. One is they need to keep good records and stay organized. I mean, in a decade, I did run across one operator who would grab the wrong bottle of standard and fail the system. But I mean, that's a pretty rare case, which is why I pointed out it happened once to me in a decade. So that's one extreme, and part of their problem is they were just too distracted by other things. So you'll need an operator that's going to get the right material into the analyzer and follow the procedure carefully, and that's it. The verification usually is not rocket surgery, but you need to be careful. That's all.
Yeah. Thank you. Anything that you can comment on microplastic specifically?
Yeah, so I guess one is, if you're analyzing microplastics, it's like analyzing anything else. Actually, the polystyrene latex are effectively microplastics. I mean, they're tiny little plastic spheres. There is a huge interest in microplastics as contaminants of the environment, which would be questions we kind of get at after the verification of the system. So once you know your system is operating, then you'll go back and run your size distribution analysis on your unknown materials. The other comment on microplastics is the folks I've run across often are interested in kind of these mixed samples. They're part what they'll call benign organic particles and part less benign microplastic particles, natural origin particles mixed with microplastics. And then you might need something like a Raman analyzer to determine which is which. But that's kind of well beyond the scope of this, but you can shoot me an email and I can put you in touch with people over here working on it.
Thank you, Jeff. So email, you can reach out to us at labinfo@horiba.com.
Yep.
All right.
Okay, I think we're all set. If you have a question later on, just send us an email.
Absolutely. Thank you. So for now, have a great day, and we'll see you at one or more of our future webinars. Thank you, everybody. Thank you, Jeff.
Okay. Thank you.